Hi everyone. In this video I am going to explain you about prokaryotic cell. Prokaryotic cells. The cells that have membrane-bound nucleus and membrane-bound cell organelles are called eukaryotic cells. And the cells which lack these membrane-bound structures are called prokaryotic cells. The prokaryotic cells are represented by bacteria, actinomycetes, mycoplasma or PPLO, Pleuro Pneumonia Like Organisms, spirochaetes, rickettsiae, and blue-green algae or cyanobacteria. The cells which lack of membrane-bound nucleus and membrane-bound structures in the cell called prokaryotic cells. Example: bacteria, actinomycetes, mycoplasma, spirochaetes, rickettsiae, and blue-green algae. Morphology of prokaryotes. Prokaryotic cells differs in size. Most of the bacterial cells are 1.1 to 1.5 micrometers wide and 2 to 2.6 micrometers length. Smallest bacterium is Dialister pneumonia. Bacteria like spirochaetes are very long, up to 500 meters in length. Epulopiscium fishelsoni, a huge bacterium discovered in the intestine of the brown surgeon fish, measures up to 600 micrometers long and 80 micrometers wide. A larger bacterium, namely Thiomargarita namibiensis, is a spherical bacterium measuring about 700 meters in diameter, was recently discovered. Longest chain-like bacterium is Beggiotoa. Prokaryotic cells lack a defined nucleus but have a region in the cell termed the nucleoid, in which a chromosomal circular double-stranded DNA molecule is located. Archaeal membranes have replaced the fatty acids of bacterial membranes with isoprene. Some archaeal membranes are monolayer rather than bilayer. Prokaryotes can be further classified based on the composition of the cell wall in terms of the amount of peptidoglycan is present. Prokaryotes lack of membrane-bound nucleus, but the region where single chromosomal circular double-stranded DNA molecule is located, that is called nucleoid. The prokaryotes are divided into different types based on the amount of peptidoglycan in their cell wall. Gram-positive bacteria or gram-positive prokaryotic organisms typically lack the outer membrane found in gram-negative organisms and contain a large amount of peptidoglycan in the cell wall, roughly 90%. Gram-negative bacteria have a relatively thin cell wall composed of a few layers of peptidoglycan. Gram-negative bacteria have a relatively thin cell wall composed of a few layers of peptidoglycan. Gram-positive bacteria are gram-positive prokaryotic organisms. Cell wall is very thick and 90% of this cell wall is composed with peptidoglycan. Compared to gram-positive bacteria or gram-positive organisms, gram-negative bacteria or gram-negative organisms contains thin cell wall and composed of few layers of peptidoglycan. Nucleoid, the irregularly shaped region within a prokaryotic cell where the genetic material is localized or where the genetic material is present, called nucleoid. Plasmid, a circle of double-stranded DNA that is separate from the chromosomes, which is found in bacteria and in some protozoa called plasmids. Osmotic pressure, the hydrostatic pressure exerted by a solution across a semi-permeable membrane from a pure solvent is called osmotic pressure. Remember, the region of bacterial cell where the genetic material is present is called nucleoid. Nucleoid is non-membrane bound, irregularly shaped region. Plasmid. Plasmids are extrachromosomal, circular, double-stranded, antibiotic resistant, self-replicating genetic materials in bacteria. Plasmids are generally found in bacteria and in some protozoa. Let's see the diagrammatic representation of bacterial cell structure. See, the very outermost layer is cell wall, which is made up of peptidoglycan. Below to the cell wall, bacteria contains plasma membrane, and these prokaryotic cells also contains flagella, which helps in motility, and they also contains filamentous structures which are called pili or pilus. Sex pili helps in conjugation. Pili helps in attachment on substratum. Within the cell, it contains a cytoplasm. In the cytoplasm, they contains 70S ribosomes. At the region of nucleoid, bacteria contains the main genetic material or chromosomal DNA, which is called genophore. Let's check this diagram carefully for your better understanding. Let's see the differences between prokaryotic cell and eukaryotic cell. The very first one, in prokaryotic cell, nuclear material is not bounded by a definite nuclear membrane. Coming to eukaryotic cell, nuclear material is bound by definite nuclear membrane. Prokaryotic cell, genetic material is made up of a circular DNA without histone proteins called genophore. But in case of eukaryotic cell, DNA is associated with histone proteins to form definite bodies called chromosomes. In prokaryotic cell, DNA content is low. But in case of eukaryotic cell, DNA content is comparatively high. In prokaryotic cell, one chromosome is present. But in eukaryotic cell, two or more chromosomes are present. In prokaryotic cells, membrane-bound cell organelles are absent. But in case of eukaryotic cell, membrane-bound cell organelles are present. In prokaryotic cell, cell wall, non-cellulosic, contains amino sugars and muramic acid. But in case of eukaryotic cell wall doesn't contain these substances. In prokaryotic cells, if flagella and cilia are present, do not shows 9+2 arrangement. But in case of eukaryotic cell, flagella and cilia contain 2 central and 9 peripheral strands, which is called 9+2 arrangement. In prokaryotic cell, cytoplasmic streaming is absent. But in case of eukaryotic cell, cytoplasmic streaming is present. In prokaryotic cell, chlorophylls, if present, are associated with lamella, which is called chromatophores. But in case of eukaryotic cell, chlorophylls are associated with chloroplasts. In prokaryotic cells, true sexual reproduction is absent. Example, bacteria, blue-green algae, mycoplasma. But in case of eukaryotic cells, true sexual reproduction is observed. Example, all animal cells and plant cells. Let's check this table carefully to know the differences between prokaryotic cell and eukaryotic cell. Up to here, prokaryotic cell was completed. In next video, I will start to explain you about eukaryotic cell. Thank you.